Hello, this is Dr. Dan Guerra again from the VerEvMed studios from the beautiful Pacific Northwest of the United States. Today we're going to talk about a topic that will require probably a little bit more background than most folks might have initially. That's okay, because I'm going to walk you through it. The reason we're going to talk about this paper, which I'm going to go ahead and get this started right away, is because this topic of autophagy comes up again and again in the literature. Remember what autophagy is. It has to do with the destiny of cells that have been programmed in a specific direction. That destination that the cells go to will then decide whether or not those cells' fate will ultimately be decided. That is, will the cells maintain their normal metabolism and their normal functions? Will the cells arrest, stop growing, and consume internally macromolecules and macromolecular and organellar structures, which is what autophagy is? Or will the cells kill themselves or, in fact, just die via necrosis? So those are various destinies of a cell in any tissue type. And the reason why that's important is because all cells ultimately die. It just depends on how, which we just described there, and when. Now, if they die prematurely and they're in important vital tissues, such as, say, in the pancreas, that's not a good thing, right? So unless you have rejuvenation of those cells, as you have new cell division, so that you get more cells in that particular, let's say, acinar pool of ductal cells in the exocrine portion of the pancreas, you're going to lose that function of exocrine capability of the pancreas. And that's not a good thing. Likewise, you can think of this happening in the brain, in the heart, liver, etc. Today, we are going to talk about ER stress in the pancreas, because that's really important, because pancreatic cancer, something that I spent a really well-described, well-detailed, and according to what I've noticed online, well-received VerEvMed lecture on ductal carcinoma of the pancreas. So today we're going to look at it from a different perspective. Today we're looking at mechanisms, right? And the mechanisms involve, again, whether or not a cell decides upon signaling whether or not it will quiesce, whether or not it will maintain, whether or not it will carry out this limited proteolysis and lipase activity so that it resynthesizes monomeric organic molecules and maybe makes a little bit of energy in the deal, but otherwise does not kill itself or actually outright kills itself. Now remember, that's really important because it's a tipping place for whether or not a cell decides to maintain its integrity or to divide. And if it divides and it's in the wrong compartment, such as in ductal carcinoma, then you get cancer, right? So it's a fine line. It's like a seesaw event, and this homeostasis is really tightly controlled. So anyway, that's me, Dr. Dan Guerra. I'm going to talk about autophagy, how it plays a role in endoplasmic reticulum stress-associated cell survival. This is my Facebook page. Please go to it. You'll see all my VerEvMed lectures there, including a lot of other things, including philosophy, mostly Western philosophy, and some pictures, etc. For fans. And of course, also, this is our website for VerEvMed. Please go to our website and hook up with us through this email address here. You can also hook up with me directly on this email and talk to me about any of the lectures that you see or any indications or questions you have about VerEvMed. We are a privately owned company that deals directly with clients, and we help go into the scientific literature, use that scientific literature as a foundation to answer questions in biomedicine, for example, but also general questions in biology and natural sciences, and also for patent attorneys and for people in the pharmaceutical industry. Okay, so that's our logo there, of course, beautiful logo. This picture here, I normally tell you where the pictures come from. This is actually right outside my door. This is the Clearwater River, and that's actually the route that was taken by the Lewis and Clark Corps of Discovery, 1804, 1805. In fact, they probably stopped around here. So that's what I get to see every morning. And again, it's a beautiful location. So enough of what great place I live. I'm sure you live in one too. So let's talk about this topic. All right. So on go the glasses. So I've got two cartoons here. Let's take a look at the one on the left initially. This comes from an Invitrogen company slide, selling you products, of course. So I'm just freely took it. Okay, now take a look at this. You've heard me talk about this target of rapamycin. This is a really important protein that basically, when it's turned on, okay, it's in a kinase cascade. When it's turned on, you're talking about anabolism. You're talking about making macromolecules. You're talking about lipogenesis. You're talking about protein synthesis. You're talking about all those pathways that could ultimately set up for that cell to be very dynamic, very active, and then ultimately, with the right signals, go through cell cycle and reproduce itself. Okay, so mTOR, when it's fully up and functional, is an anabolic cell when that's turned on. Right? It's called mTOR because it's a target of an antibiotic called rapamycin. So if you add rapamycin, when you see an experiment that adds rapamycin, it's blockading the mTOR. mTOR is a complex, that's why you see a little C there, and there's a couple of different complexes, mTOR1 and mTOR2, and 2 talks to 1 and reactivates it or hyperactivates it. That's all you need to know for right now. Want to know more about mTOR? Just go back to some of my previous talks. I've gone through detail on this again and again and again. All right, so let's take a look at this. In times of nutrient abundance, this is any given cell, mTOR is turned on, okay? When it's turned on, look what it does. It blocks this whole pathway. Now, this pathway down here, the ULK, the ATG, whenever you see ATG, think of autophagy, FIP200, all of that complex will then invite the ATG9L into this new complex, which is also called the phosphorylated kinase 3 complex. And that will start the process of autophagy in that cell. So if you block mTOR, so nutrient abundance, this gets blocked and this doesn't happen. But if you block mTOR, this does happen. Okay, that's how these slides go. I'm sure most of you know this, but for those of you that don't have a background in these kinds of diagrams, you need to understand that that's a blockade, and the arrow means yes, go on, right? Okay, so let's take a look here. Nutrient excess, mTOR on, blocks this whole pathway. We could just wipe it out of that slide. And I told you that mTOR just basically means anabolism, protein synthesis, lots of activity and lipogenesis, things like that. Now, there's all kinds of things that block that. But in physiological terms, AMP kinase blocks it. Now, AMP kinase, again, I talked about it in previous VerEvMed lectures. That's adenosine monophosphate kinase. Remember, kinases add phosphate to proteins, so it's a protein kinase. Now, that protein there, it comes up, it builds up activity when you have a lot of AMP. You have a lot of AMP because you've exhausted the ATP. Remember, it goes ATP, ADP, AMP. Okay, triphosphate, diphosphate, monophosphate. And as you strip off phosphates from ATP, you get energy, right? So this is a cell that's in stress. When it's in stress, it often is because of nutrient depletion, but it could be all kinds of other reasons, right, why a cell is in stress. So this is like a major player in cell stress. AMP kinase then blocks mTOR. Makes perfect sense at the logical level, okay, at the level of square of opposition, right? All of this or none of this. You do not want anabolism when the cell's in stress. You don't want to be spending all this metabolic energy making proteins when the cell's in stress. And it has to decide, or its decision is made for it to maintain, to autophage, or to kill itself, or ultimately, again, to divide. Okay, so the AKT pathway, of course, with growth factor stimulation, insulin's all going to be positive mTOR. Okay, so we're not going to talk about that today. I have in the past. Because this comes from Invitrogen, they're telling you all these really cool pharmaceuticals they have and drugs and research tools that block and turn on. So that's all these things are here. Don't worry about them. So metformin is interesting because metformin, of course, is a drug used in diabetes. Okay, and so the reason it works is, we talked about it before. I'm not going to go into detail. There's resveratrol. Wow, that polyphenolic compound that gets a whole lot of press time. And then it's very well received in the scientific literature. It actually blocks that pathway too. But I'm not going to, and that has to do with histone, acetylation. Resveratrol is a very complicated compound, but I've talked about it again in a previous lecture. Look it up and you'll see. Okay, so let's finally get to the autophagy lysosome synthesis. This whole protein synthesis is turned on because of AMP, because of stress. ULK, ATG13, FIP, and ATG101. Pick up a recruit 9L. You convert this whole complex here, this complex and that complex together, take a phagophore, which is just a membrane, and picks up a whole host of other autophagy proteins, and then works with this linear peptide of another membrane-associated protein called LC3, which is an ATG-associated protein. And you start to curve that membrane and isolate components or members of the cytoplasm at that time. Whatever's in the cytoplasm starts to get engulfed. Ultimately, that entire phagophore closes. Now we're going to call it an autophagosome because it's going to carry out autophagy. Now, it also has to fuse with the lysosome. You know, lysosome is a compartment in the cell that deals with degradation. So it has all the degradation enzymes, proteases and glycosylases, lipases, things like that, right? RNases. So when you get the fusion of the lysosome with the autophagy lysosome, you start to disintegrate. That is, break down entire organelles because the membrane can be pulled apart, and all the macromolecular structures like polyribosomes. You can break down the endoplasmic reticulum, the peroxisome, in fact, the whole nuclear complex, right? And that's now called the autophagy lysosome. That's this structure here. And ultimately, everything gets degraded. Now, remember, this is a limited thing. Notice this phagophore just picks up a section of the cytosol. So only that part of the cytosol that gets treated with this digestion, remember that autophagy, phagy means to eat, will be broken down. It's not broken down to the point that it's no longer used. Again, it's broken. The proteins are broken down into individual amino acids. The lipids are broken down into fatty acids, isoprenyl units, glycerol, sphingosine base, and all those are signaling molecules, and they can also, be reused. So it's not like the cell is killing itself. Now, if this proceeds and is chronic, it can lead to another stage, and that would again be frank cell death, right? It can go that route. This is not the cell death pathway. This is autophagy. It's a different thing. It's a maintenance control mechanism during stress to digest what you have, kind of like freeze what's going on, make the building blocks for new proteins, new lipids, new RNA molecules, new DNA molecules, eventually, so that you can turn things back on when the stress is removed. That's what autophagy is about. Really, really cool cellular mechanism. Okay, so here, this slide here basically tells you what it's about, going from the beginning of autophagy all the way to where everything is degraded. Now, autophagy has been implicated in pathophysiological and in physiological functions. So all over the map, as you might guess. It can lead to cell death. If it's corrupted, it can lead to cancer. If it's overturned on a chronic, it can lead to cancer. It can be a direct conduit to the innate immune system, such as macrophages, eosinophil complexes, all of the innate immune system, some of the natural killer cell systems. Autophagy has been implicated in cardiomyopathy. We talked about that in a previous talk. Aging, I talked about in a previous talk. Infectious diseases. That's going to be a next big topic we're going to do in 2018. So hold on the fort on that one. I'm going to start talking about infectious diseases because it's another one of my passions. It seems like I have passions for pathology. I don't know why that is. Probably my training. Hopefully, that's all it is. Okay, autophagy is also, as you can see this metformin story here, involved in type 2 diabetes, fatty liver, right, which is all not good, and neurodegenerative diseases. Like Alzheimer's and Parkinson's disease, autophagy can play a role, positive role, negative role, depending on how it's regulated or dysregulated. So it's a major player in biological systems, and particularly in biomedicine. Now, that's macro autophagy or total cellular autophagy. You also have micro autophagy, and that includes mitophagy, pexophagy, and ERphagy. Sorry about that last one. I didn't coin any of these, by the way. Now, mitophagy has to do with the self-consumption of mitochondria, and only the mitochondria, thank you. Pexophagy, self-consumption of the peroxisome. Very important, very cool organelle, particularly for your lipid biochemists like myself. And then unbeknownst to you that are not lipid biochemists, it's still a really important, really cool organelle. It doesn't get enough attention. But that's an organized destruction just of peroxisomes. And then there's the ERphagy. Of course, that's endoplasmic reticulum. And that's the topic of today, and that's why it's highlighted. All right. So let's get through this, please. Autophagosomal membranes form de novo, okay, so they can form just from lipid precursors or from the endoplasmic reticulum and or the mitochondria or the Golgi immediate compartment. That means those compartments which are more cis into the cell rather than trans. Or indeed even the plasma membrane or from endocytic endosomes coming from outside the cell. So the fragments of membranes can be used to make that initial phagophore that we saw 2 slides back. Now, ATG, or autophagy proteins, cluster. So you have the ULK, which is the uncoordinated 51-like kinase. I just dig that name, uncoordinated 51-like. Wow. I wonder what the 51-unlike kinase is doing. Well, I don't know, actually. Probably doesn't exist, though. In any rate, because then it would just mean you wouldn't need to have all those adjectives, right? Yeah, modifiers. All right. Anyways, the ULK complex is composed of, it's a kinase, see the big K there, and you have ULK1, 2. You also have scaffolding proteins, and these are proteins that help organize the structure around the membrane, the phagophore membrane. HG13, FIP200, which actually stands for FAC, interacting protein 200 kilodalton. Boy, that's really helpful, right? But anyway, it's a kinase, that K. And then you have ATG101. Right? So what happens is ULK phosphorylates ATG proteins, among lots of other autophagy proteins. Ubiquitin-like ATG8 proteins of the LC3 and GABARAP subfamily, these are still all in the ATG superfamily, are recruited to those phagophore membranes via the C-terminal lipidation, which facilitates vesicular closure, or that is the phagolysosome closes. Now let's look at this cartoon over here. So you have growth factor signaling, stress, energy, nutrients. This is normally what you're getting with a happy, healthy, well-fed, maybe overweight state. mTOR is on. All these great things happen. Anabolism, maybe unfortunately cell proliferation. But that's not what we're talking about today. We're talking about that not happening. So if you block it with rapamycin, or you deplete the cells in cell culture with amino acids, or all kinds of other ways, just upping AMP because you burn a lot of ATP, mTOR is gone because of AMP kinase. Because of that, it's not blocking autophagy. Autophagy then pursues its ultimate end. So the protein kinase complex, ULK1, 2, FIP200, ATG13, we already said. ATG9 is a multi-spanning membrane protein. Don't worry, you're going to see all this in a really cool cartoon soon. And you also get this lipid kinase complex. Now, what that means, it's kind of an odd name for it. When you first see it, you think, is it phosphorylating lipids? No. Actually, it's a complex of those proteins, the VPS34, Beclin, and ATG14, which are bound covalently to a lipid embedded in that phagophore membrane. It's anchoring or tethering those proteins. And when they're tethered, now they become a kinase complex. So it's called a lipid kinase complex. I'm sure you dig it. All right. Now, when all that stuff happens, so autophagy's on, a couple of other proteins come into play. There's the phosphatidylacetol 3-phosphate, etc. You recruit all these other proteins, ATG16, 5, 12. You get lipidation and membrane association of this linear peptide C3, and all these other proteins come together. That expands this whole phagophore complex. The phagosome starts to curve around. Again, that's a biophysical phenomenon because the proteins are bound to, particularly a ethanolamine, phosphoglycerol in the phagophore membrane. And when it does that, it stretches that phagosome, that autophagosome, and starts to turn it onto itself. So this is basically like closing it because proteins are knitted into, covalently bound to, these particular phosphatidylethylamine phospholipids, and also to phosphatidylinositol phospholipids in the phagophore, thus creating the biophysical phenomenon that ultimately leads to closure. And so that's just the same thing you saw before. That's just generating the autophagosome. Okay, and that's some of the more detail of it. All right. Now, this is all kinase control. So you know that AMP kinase, I already showed you. When it phosphorylates this complex, that whole complex phosphorylates mTOR-Raptor and shuts it down. Okay. Now, if mTOR-Raptor is functioning well, it phosphorylates the complex and shuts it down. So this is like repealing the autophagolysosome edict, right, from the emperor, right? using an ancient Rome metaphor. Okay, so, and remember the AMP kinase shuts down mTOR but turns on this whole complex, right? And phosphorylation of the AMP kinase itself can block AMP kinase. This is just feedback inhibition. So that's a slide I like. I got that from this 2015 paper. There's the whole citation. All right, now check this out again. AMP kinase phosphorylates ULK. Remember that one? It's in association already with HG13 and FIP200. Okay. Now, remember, you can block all this, but we're not doing it now, right? This is all turned on, and that's going to phosphorylate and then turn on and recruit all these other proteins. Okay, there you go. Okay. Now, see, I told you you'd see this really cool cartoon, and there it is. So these are the phosphatidylethanolامine lipids. There's the phosphatidylinositol 3-phosphate lipids, and these are just part of the membrane complex of the phagophore. Remember that membranes are made up of lipids. Phospholipids, sphingolipids make up those amphipathic molecules that allow a membrane to take on, for example, a bilayer like it's shown here, but it can take on all kinds of other structures. Remember, the membrane then limits the cell, right, and creates life, actually, because, of course, all life is based on lipid biochemistry. I know you all know that. And this is a good opportunity for me to bring it up again, because everything would not happen if you didn't have this membrane charged with these particular phospholipids. They happen to be glycerol lipids, so that they will covalently bind to these proteins, right? So when they covalently bind to the proteins, you basically are making a macromolecular protein lipid complex, which is going to act like a mother load kinase, right? That's the lipid kinase they talked about. Okay. So let's talk about it. This comes from Essays in Biochemistry, a paper in 2013, and this came from an ENBO journal paper, that particular cartoon. So autophagy is initiated by the engulfment of cytosolic components in that crescent-shaped structure called the phagophore, which now should be in your mind, expands and fuses to form a closed double membrane vesicle called the autophagosome. Autophagosomes are targeted then to the lysosome. They fuse to the lysosome. Remember, the lysosome has all the enzymes to break down what's in the phagosome, and so boom, you're done. You're getting into autophagy right then and there. Fate decided, bring out the tombstone for those proteins and those lipids and those nucleic acids and those organelles, right? So ATG8s, it's a whole class of autophagy proteins, are actually ubiquitin-like proteins that play an essential role in the autophagosome biogenesis. There are several mammalian homologs grouped into 3 subfamilies. There's the LC3, which is a light chain 3. There's the GABA-RAP, love that, which is gamma aminobutyric acid receptor associated protein. So yeah, does that play a role in neural synapses and turnover of synaptic proteins and changes in signal transduction cascades? Yes, it does. And then gate 16, which is the third of the superfamily of the mammalian homologs of these ATGs. Okay? And GATE means Golgi-associated ATPase enhancer of a 16 kilodalton polypeptide. Don't you just love the name of these proteins? I actually do. So the ATG8s, that's easier. The ATG8s are synthesized as cytosolic precursors. Okay, so they come from, they're not mitochondrial genes. They're actually genes that are found in the nucleus, like most of them are. But they undergo a series of post-translational modifications, and that's what targets them into this system. Right? So there you go. You've got ATG3, ATG12, and you notice you get a whole dimer here. So everything is duplicate. It's like mirror to mirror, right? You have 2 ATG21s, you have 2 3s, you see 2 12s, and this whole thing forms like this bridge complex associated with these really important ATG8s that are bound to the phosphatidylethanolامine lipid, which is embedded in the phagophore membrane. So that's got to be there. That lipid's got to be there, as do these PI3Ps. Okay. So, the paper we're actually going to talk about today is from Developmental Cell. Now, if you've never read this journal before, for the most part, it's behind a paywall, but every once in a while, it's open. This particular paper is open and free to the public, and there's the link. So you can read the entire paper, download all the figures. I'm only going to briefly go through a few of the research points in the paper. As all developmental cell papers and cell papers in general are, it is an excellent paper full of data, full of evidence that supposedly, hopefully, supports what they're saying in the end of the paper. Okay, very well done paper, by the way. I really dug reading it, and I'm glad I'm presenting it. Okay, so that's the paper. Now, Particularly this little inset I got from Essays in Biochemistry from a 2017 paper. So I took, this isn't from that paper, this is from another one, but it allows me to basically demonstrate to you with visual cue what this over here is saying, which is what I do in my talks when I give lectures too. ATG proteins facilitate cargo recognition during selective autophagy. So that's one of the things they're involved in. Okay, we didn't talk about some of their functions. We just showed you how they interact with one another. But that's basically what they do. Not all cargo is going to get put into that autophagosome. So the ATG proteins, because they are proteins, they're going to be protein-protein interactions. So you can guess there's a whole lot of things going on in terms of signaling and communication during the synthesis of the autophagosome complex in a cell that's been triggered to go into autophagy. Okay. So the mammalian selective autophagy receptors, SAR, is a cargo receptor. And the bridging of cargo to autophagy machinery occurs primarily via the binding of ATG8. And we just saw that, right, with the PE, the phosphatidylethanolamine lipids, and the phosphatidylenosyl-3-phosphate lipids and all that. ATG family binding uses a linear peptide motif known as the Lear linear peptide receptor or the LC3 interacting region. That's Lear. LC3 interacting region. Wow, cool, right? All right. Now, that R is for region, not receptor, by the way. Okay. UPR. Now, here's another concept, I guess, and I know that those of you that are students of cell biology, biochemistry, cancer biology, probably some pharmaceutical sciences, certainly vet scientists know this stuff as well. Plant scientists know this stuff. But UPR is unfolded protein response. So what that basically is when you get an unfolded protein. Now, proteins naturally fold because of their primary structure, that is their amino acid sequence. They form secondary structures, tertiary structures, and if there are multiple subunits, they make quaternary structures. So that folding process is facilitated in certain organelles like the ER via covalent modification of the polypeptide. The ER is a grand place to add sugars. So there's adenylyl pyrophosphate pathways in there, and that's where you can make glycoproteins. The ER is well known for making glycoproteins. Glycoproteins then fold in a certain way, a certain symmetry, axis of symmetry, in 3 dimensions in real time. And when they do that, they're ready to be packaged, for example, from the ER to the Golgi cis, to the Golgi trans, fusing to the plasma membrane. And if they're going to be secreted, like, say, an immunoglobulin, or let's say an interleukin, then they get free passage out of that plasma membrane because they've been correctly folded and processed, and they're going to be exported out, right? Very important thing that all cells do. It's a way for cells to package things and send them out, not only to get them out and to do something to the rest of the system, because they've been stimulated to do so. Let's say it's a T cell or macrophage or something like that, and sending out cytokines. Not only that, it's also because they need to be basically signaling or telegraphing what's going on with them, because all cells have to know what all other cells are doing, particularly the surveilling immune response, to know whether or not all cells are happy and doing what they're supposed to. You start getting mixed signals from cells, the immune system notices it, goes and checks it out. It can maybe fix it. For example, stress, autophagy. Let's tune you down, cell, make sure that you're doing okay, and let you tune back up and become the cell you were before, or knock you down and kill you, maybe even with natural killer cells or cytotoxic T cells if it's necessary, right? Or, of course, through the wonderful programmed cell death of apoptosis. Okay, so UPR, unfolded protein response, involves transcriptional mediation to correct protein structural deformity, and that's all going down in the ER, as I said, endoplasmic reticulum lumen, that's inside the ER. The UPR uses endoplasmic reticulum integral membrane protein sensors, and here they are: IRE1 alpha, ATF6 alpha, and PERK. I've also talked about this whole system in a previous VerEvMed lecture, so you can go back and look those up. Look up my name, look up those acronyms, and you'll find it, I'm sure. I've talked about it more than once, actually. Okay, so those are all sensors, and they trigger cascades that inhibit protein synthesis. Remember, that's the whole mTOR, shut you down. We don't want any more proteins being made, while at the same time enhancing transcription of chaperones, oxidoreductases, ER-associated degradation proteins. These are the ERADs. Okay, so ERAD is kind of like the UPR response once it gets going. Okay, so ER-associated degradation is just a generic term of what the UPR tells you more specifically. Like UPR tells you more about why, and ERAD tells you more about what, right? Degrading UPR because things are unfolding together. I like that. All right. UPR then stimulates autophagy, via transcriptional upregulation of what genes? Of course, the ATG genes. What else, right? ER-phagy is the autophagic sequestration of fragments of the ER into an autophagosome. Okay, so ER-phagy, the topic of today, is ER-phagy. So this is an autophagic sequestration of the ER complex, not of the whole cytoplasm. Okay. Glasses back on so I can take a look at this slide. All right. Triggers for ER stress include calcium perturbations, excess unfolded protein, okay, hyperaccumulated proteins, redox potential changes, like a whole lot of reactive oxygen being made, not being quenched, or all kinds of pharmacological inducers, right? So this is, the triggers we are talking about, pulling out nutrients, pulling out amino acids, you name it. So you get ER stress because the ER is a really important organellar system. It communicates with the nucleus on the nuclear envelope. It communicates with the Golgi apparatus. It communicates with the endoplasm, with the mitochondria, with the peroxisomal system, with the ribosomal system. It's got its own ribosomes. It's got its own polypeptide synthesis. It's got all of this post-translational modification, including glycosylation and lipidation. So ER is really an important sort of like completely involved organellar complex in the cell, right? ER is really super important. Okay, so you could either go through the overload response, endoplasmic reticulum overload response, and that triggers a different thing. Overload means there's just too many things happening. You get an NF-kappa B, and that does is that induces the production of pro-inflammatory cytokines. And it can also, in fact, depending on the right distal signaling, cause cell proliferation. So this isn't a very good pathway, right? Because it's going to cause inflammation, and chronic inflammation can lead to, tumorigenesis, or it can lead to autoimmune responses. So this is a dangerous pathway, but it can be turned on and necessary at times. Likewise, proliferation can be useful at times, of course. But we're not talking about that. We're talking about the unfolded protein response. And here you've got the three main proteins, okay? The PERK, the IRE1 alpha, and the ATF6. All of those then work together to give you an outcome. Now, what's the outcome? You can get just direct proteasomal ERAD, okay? That's ER-associated degradation, as we talked about before. Degradation of what? Those organelles, the proteins, the ribosomes, all that. You can also get just an increase in ER volume. Why is that important? If you increase ER volume, you're going to decrease the relative concentration of those sequestered macromolecular structures, which are trying to precipitate out. I bring in the word precipitate because that's going to be important when we look at the data here in a moment. So when proteins start to precipitate, that means they become not soluble in, say, the ER lumen, but they become solid in the ER. When they become solid, they can't be reacted with very well, and they need to be eliminated, right? Because large complexes are not what you want inside anything, especially inside a cell. The outcome can also be an increase in redox regulation. I mean, get rid of those reactive oxygen species, which are always generated during stress. Because of basically uncoupling of the electron transport chain, and just a whole lot of iron free and copper free and free radicals, and just getting rid of electrons. You can actually also go through apoptosis. So the UPR can lead to apoptosis, and I talked about that in the previous lecture. Or as today, ER quality control autophagy. This is kind of like the refined type, right? The kind of like Victorian answer. Oh, gee, there's something going wrong here. Let's deal with this ER stress, but let's be very specific. Let's deal with only certain proteins in the ER. Quality control. A little bit too much of this, get rid of it. Not enough of that. Ultimately, turn around, try to promote more of it. ERAD3, which again is a degradation pathway, ERAD2, that's a unique one, not as well characterized as regular ERAD, but that also is a limited degradation pathway, or the ER-phagy, okay, and we're talking about here. All of these are actually autophagy, okay? They go through the same basic thing, the phagophore encircling, right, the phosphatoethanolamine, the phosphatidylanoxidil binding to those ATG proteins, and their linear sequences associating with that lipid kinase and all that. All right, so you get the situation. So you start with stress induction, signaling, and then effection. Okay, that's the way everything works in the cell. All right. There's just another way of looking at it. I like this slide, so I put it in here. 2015 paper in Nature. Now, this shows you there's two different kinds of phagy. There's the one that deals with perinuclear ER, nuclear envelope, because remember the ER is in association very intimately with the nuclear envelope. So when you have ATG39 turned up, that's an ATG39 dependent perinuclear phagy, nucleophagy. That leads to autophagosomes generated from the nuclear envelope proteins, as well as the ER associated with it, popping into the vacuole and getting ready to go to autophagy. That is in contrast to the normal cortical ER, cytoplasmic ER, which is non-nuclear associated, so it's not perinuclear. That's an ATG40 dependent pathway, and you either have the cytosolic or you have two different forms of the phagy. Okay. And that ultimately leads to the same thing, what we're talking about, phagy. Okay. So, all in the FAM, because we're talking about FAM proteins here. There are at least 3 phagy membrane-anchored cargo receptors, FAM134B, RTN3, and SEC62. So the wild-type FAM134B is required for maintenance of peripheral sensory neurons. That's how it was first described. So if it's involved in peripheral sensory neurons, you know that if something goes wrong with it and it starts turning on autophagy, the cell's destiny is going to be altered, maybe incontrovertibly. So that happens in the peripheral sensory neurons. What about other organs and cells? Well, today we're trying to lock in on acinar cells. Those are ductal cells of the exocrine pancreas, not the endocrine pancreas. We're not talking insulin glucagon here, somatostatin here. We're talking about those enzymes that are secreted by the pancreas that help in digestion, okay? Lipases, glycosylases, proteases, that sort of business. All right, so that's what the acinar cells do. Now, I think you might remember that the acinar cells are the ones that really get, when you talk about pancreatic ductal adenocarcinoma, right? PDAC is at the acinar cell, right? It's not with the endocrine pancreas. It's with the exocrine pancreas. That's why it's much more common. That's why it's so deadly. Right? All right. So acinar cells of the adult exocrine pancreas secrete proteins called zymogens. Zymogens are digestive enzymes, but they're not yet activated. Not activated because if they were activated, they'd be digesting everything along the way, including the acinar cell. Not a good thing. In fact, rather asinine, right? So they come out as zymogens. It's an old term, which just means an enzyme that's not functional until it gets covalently modified, usually proteolytic limited degradation. Zymogen can convert it into a regular enzyme, and when it does so, it becomes a pancreatic digestive enzyme. Okay. And then again, you have to take care, keep as zymogens when you're moving through the pancreatic duct, which is what is composed of the acinar cells. So a balanced unfolded protein response is critical for pancreatic acinar homeostasis. Sorry, I didn't put that. Why that's there, I don't know. Mistake in the typing. So why is that? It's because, remember, you're making a lot of zymogens when the pancreas is functioning, which is like all the time, but particularly when you eat. And if you eat a lot, more, right? So diabetic state, overeating, obesity, yeah. That's why some of this can be linked back to pancreatic cancer. It can. Not necessarily, but it can. There are good theories about that. Anyway, because the pancreas is, the acinar cells are basically ducts that are releasing a lot of protein. Those proteins can be overproduced. When they're overproduced, you've got to get rid of some of them because they're not all folding correctly, because they're not all being glycosylated correctly, or zymogen formation is somehow inappropriately organized. So inhibition of general autophagy by the knockout of 5 or 7 can lead to ER stress, including the cessation of zymogen protein production. So you get none of it coming out. And that can ultimately lead to apoptosis and in fact, also necrosis and inflammation. None of this is good, right? Because this could be leading then along the way to pancreatic cancer. All right. So what they're going to present here, identification and characterization of a new protein on the block called CCPG1, which stands for cell cycle progression gene 1. And we're going to find out it's a transmembrane protein. Remember all those other transmembrane proteins, all those ATG complexes. It has dual affinity for the GABA-RAP LC3 and FIP200 binding autophagy. that drives phagy. So this is a new player that seems to be necessary for that whole complex to come together at that phagophore. Okay, so they were looking for what is the proximal signaling molecule that's going to start this process as you're starting to form that phagophore, starting to close that structure. So it was discovered that this CCPG1 was the player, at least one of the players. It requires ATG to make this whole thing happen via two discrete interaction determinants. CCPG1 prevents accumulation of insoluble protein, which is good because insoluble protein is bad, within the endoplasmic reticulum lumen, thus sustaining the exograft function. So CPG1 is necessary, essential and necessary, for this whole process to keep that UPR functioning again at some homeostatic level. So not so much in pathophysiology, but in normal physiology, this protein's functioning as well. That's the important thing. All right. So there's our first real data. Okay, take a look. Glasses on. These are pull-down assays, Western blot, immunoblot. First of all, take a look at the protein. They're describing the protein as having an N-terminal domain, which is cytosolic. It's inside the cell. Then they have the luminal portion, which is in the lumen. Okay. So of the membrane, and there's a transmembrane region there. So that's what the protein looks like. So a lot of the activity we're going to talk about today is the NTD, the N-terminal domain of the polypeptide. And remember, this is CCPG1. So what you've got here is you're looking at antibodies raised to this MYC CCPG1, and you've got that structure being used as bait by the antibody that normally recognizes GST fusion. So you have a glutathione transferase fusion to the CCPG1, basically. So you've got a protein that's going to recognize that because GST is bound to it. Okay, you've bound this protein to it. So if you look at the reaction with GST, okay, low GST input. Now this is a crude lysate. What do you get? You get a response for GST, right? And you get a response at the level of the C. So if you're using GST or CCPG and running in GST alone, you get both a reaction to this structure, antibody to this structure, and the antibody to GST. Likewise, if you're using LC43, you get something. You use this second linear molecule, you get something. And with the GABA-rep, you get it. All of these are noticing this antibody to this antigen. Okay, so that means that you're getting a protein-protein interaction, or you're pulling down those proteins. So that says CCPG pulls down all the affinity purified, we're going to get to that in a moment, of the ATG8 family proteins, LC3B, LC3C, and the GABA-rep. Now, check this out. Okay, look at this. If you have just the antibody and you affinity purify now the GST, right? This is the GST fusion. You're not going to get any activity, right? Because you've purified it. So this is not going to react with that portion of the molecule because it's not there. It did before you purified it, but not afterwards. Yet look at how strong the signal is with these three proteins, 3B, 3C, and the GABA. What that tells you is when you affinity purify, basically you're isolating, you're distilling the response. You get a whopping protein-protein interaction when it interacts with CCPG1. That means that every one of these ATG-type proteins are really specifically interacting, being pulled down by this probe here, the CCPG1, the antibody to that. Now look at the GST profile. The GST profile, of course, you're still going to get a lot of binding to GST, is going to be as the probe. But look what else you get, okay? You get this whole myriad of proteins with affinity purified GST. That means that there are a lot of things going on with GST, glutathione transferase, and these polypeptide complexes, okay? Which, again, you would probably expect. Now, here's the more important thing, more important data point to me. CCP binds to cytosolic N-terminal domain region, but mutants don't bind. So again, here you've got input where it's crude. This is just the MYC CCPG N-terminal domain, right? Of course it reacts with GST. Of course it reacts with GABA-rep. Of course but whoa, it's reacting with this mutant here of GABA-rep. Now it's not supposed to because that N-terminal domain has been corrupted. So you shouldn't be getting that, right? And sure enough, when you affinity purify, that's what this is, affinity purify using GST, using glutathione affinity purify. Look, the stuff that's just the CCPG that's just associated with the MYC complex, wow, yeah, you're reacting with GABA-RAP. No, you're not reacting with the N-terminal disrupted GABA-RAP. That tells you the N-terminus of GABA-RAP is what's interacting with CCPG1. And this is just the GST control showing you that you still get reaction with both of these because it has nothing to do with the N-terminus of that molecule of CCPG1. That's all that's saying. Okay. Now, this is just showing you where you get specific association of these proteins with the lipid moiety, and those are the amino acids involved. You can see that all different systems all have this motif. Okay, all have a motif. Okay, so and you can see that it's quite well evolved, right? So that, again, not as important as this slide here, or this portion of the slide here. Now we're looking at input again. This is not purified. Now we're looking at a his-tagged GABA-RAP and whether or not it picks up GST or it picks up the GST fused to CCPG1. So there's GABA-RAP. So you're getting it when you add, right, this protein complex. You get this immunoreactivity. But now look when you affinity purify. Holy smoke. You get a whole large amount of interaction. This tells you that GABA-RAP really reacts intensely with CCPG1. That's all it's telling you there. Okay? Again, very important. Again, these are just different species showing you that it's evolutionarily conserved. That's what I was trying to think about saying before. Evolutionarily conserved. Okay? Mutated Lear motif, inhibited binding of GABA-RAP, and LC3BG. Okay, and that's the G here. So when you mutate the Lear motif, again, what happens? You lose, when you affinity purify, you lose the reactivity. So you need that N-terminal, right? That N-terminal of that Lear motif of GABA-RAP is absolutely necessary. That's what this is showing you. See, you get no reactivity when you got the mutant here, only when you have the parent protein. And GST, of course, doesn't care because it's not recognizing that subdomain. Summary of the slide is CCPG1 directly interacts with ATG8 family proteins via the canonical LIR in the cytosolic N-terminus of CCPG1. That's what all of this data basically says. Now, this comes from doing mass spec work and running a computer program to look at what proteins interact with CCPG1. And they developed two scores that you get by doing mass spec analyses. There's the Z and the WDN. Now the difference here, there's differences in the way these scores are done. These are based on algorithms. But the Z score determines the number of standard deviations away from the mean. That means the more and more you get away from standard deviations, right, the more and more you're purifying for a real interaction, right? The caveat, however, for this Z scoring is that you use the MS scoring for sure, but it tends to upweight unique proteins, no matter their abundance. So it doesn't tell you really about the quantity. So you know that it's happening. That's really important. You get the quality, you get the discrete interaction because you're looking, in fact, for those rare proteins, which is what you're worried you might miss, but it's not telling you abundance. The WDN score allows for this. So only if a frequent interactor displays the observed characteristics of a true interacting protein with CCPG1 would you get enough weight factor, and that means you get the thicker line. So that's the WDN score versus the C score, okay? So again, you get CCPG1 interacting with two different classes of protein complexes. The ULK complex, which is what we're arguing that helps associate these proteins into that phagophore membrane. And then, very interestingly, all these RNA biogenesis proteins, okay? These are all going to be involved in controlling small interfering RNA, going to be controlling the processing of messenger RNA. These are really important proteins, right? So, again, you have to keep in mind that the fact that the CCPG1 is interacting with these different proteins means that it is a central proximal player in the whole control of the phagosomal genesis, and then ultimately to the autophagy associated discretely and specifically with endoplasmic reticulum. So the summary of this data, you don't see the data, but basically the data is in the spreadsheet, so I'm not going to show you. But I did look at it, and this data is indicative of what the spreadsheet said. Summary is that ULK complex, boom, and RNA biogenesis proteins, boom, interact quite well, thank you, with CCPG1 using 2 different scoring types. Okay. All right. Now, here's another interacting system, again, another immunoblot, where now you made a fusion with P flag. Here is input just coming directly from the P flag purified, and then you have immunopurified. So this, again, is an enrichment for the fraction. So the cruder fraction, the input, you react with or without using the P flag. Okay, it doesn't matter. You get binding all the way down, except maybe with the CCPG, you get a little bit less when it's not there, but you still get some. But certainly you can see that CCPG, recognizes CCPG with the antibody. That makes sense. But this is a bad set of data. It's supposed to have a band over here. In fact, when you immunopurify, which is what they do here, that non-specific banding goes away. Whenever you don't have this P flag CCPG antibody there, right, you don't have that particular bait there, you get nothing. When you immunopurify, you get interaction with 200, with HG13, ULK, AG101, all the players. But now look, you get to AG5. Now AG5 is one of those general centralized autophagy proteins, not specific for the endoplasmic reticulum. And look, you get nothing. So that means that CCPG1 is not involved in general autophagy, only in ERphagy. And that's the whole point of the paper. So CPG interacts only with ERphagy proteins and not the general ATG5. Okay, should be a G there, not an F. Okay, so that's important. Now, last bit of data slide. Glasses back on because it's all kind of small. All right, they wanted to investigate CCPG1, and so they generated a mouse, which they call a hypomorphic gene trapped. That means they trap this gene so it can't get expressed. So there's the wild type, and there's the gene trapped, right? And you get hypomorphic because it means that you're controlling the overall expression of this trapped protein. Okay, so it's below the normal, so it's hypo. Look at this. When you have the wild type, you get plenty of CCPG. When you trap it, you get nothing. Does that the same with this just, standard marker for protein in a cell, tubulin? No. Tubulin, of course, you pick up in the antibody plot no matter what. So what does that tell you? That this particular gene trap targeted CCPG1 and not other proteins. This is just taking this data, just showing it to you on histogram. Big statistical difference. Basically, there's nothing there, right? So when you gene trap CCPG, you basically sequester it so it can't be recognized, so nothing is working appropriately now, right? You don't find it. So that's a control experiment to make sure you have a hypomorphic mouse model. That's now a gene trap mouse model for that gene. That's the whole reason for that experiment. Now, C is kind of interesting. These are animals, and you're looking at the pancreata of a wild type animal and of a gene trapped animal. Notice the 6-week-old CCPG1 deficient mice revealed abnormalities. These tissues appearing whitened, I don't know if you can see that there, and opaque relative to control pancreatus. So a big, huge effect on the pancreas because of eliminating, or that is trapping CCPG. So that is tremendously bad. That's why, again, it's morphic, because it's altering actually cellular structure. So the acinar cells and the entire organ, the pancreas. So what they did is they took the pancreas from both the wild type and the gene trapped. They spun it down in 4% SDS, that's a detergent, sodium didecyl sulfate, and they get a soluble and insoluble fraction, okay? You can see that there's a lot of insoluble in the gene-trapped one, okay? And when they wash it down again, you get insoluble protein. Now, what does that mean? 8-mol-urea is going to break that down, so you don't see the insoluble anymore. Why? Because urea obliterated it. Dithialthreatol will break any of the disulfide bonds. So you get rid of that. That's a way to show you have an insoluble protein. You bang it with 8 molar urea and 1 or 10 millimolar diethylethreatol. None of those bonds are there anymore. It denatures the protein. No more insoluble. A trick in the test tube, we call it. Microphage tube, actually. All right, now this is a heat map. Okay, so this is basically showing you that when you gene trap, you get specific proteins lighting up. What are those proteins? Gene trap, wild type, gene trap, wild type, gene trap, wild type. 3 different experiments. You're lighting up specific proteins, and the proteins you're lighting up are the proteins involved in the secretory pathway of the exocrine pancreas. So, first of all, let me go through what this says here. Opaque mass could not be soluble, strong detergent. LC mass spec was performed to analyze the material enriched in the insoluble fraction. That's 6D. That's the heat map experiment. Top rank protein species differing in solubility between 2 genotypes to identify. So these are the ones you're looking at that are insoluble. Okay, now that's E. So let's take a look. Wild type, soluble, insoluble. You don't have much insoluble in wild type. Why? Because you're not screwing up the autophagy system. You're not gene trapping CCPG1, which is an important player in normal physiology. You trap that protein, look what happens. You get some insoluble, right? And this is the BIP, this is the chaperone protein for proteins being translocated, cargoed into the ER. Trypsinogen, another really important protein. Holy cow, you make a lot of insoluble when you don't have the CCPG1, which is allowing you to get rid of insolubles. Amylase, these are secreted proteins, by the way, from the exocrine pancreas. Trypsinogen is a protease. Amylase, which is a glycosylase. CBPA, okay, that's the carboxypeptide. That's another protease. And there's alpha tubulin. Okay, now look at this. Holy smoke, lots of insoluble trypsinogen when you don't have CCPG1. That means the autophagosome is not working. Likewise, what happens with trypsinogen? You get insoluble, right? You didn't before. Amylase, insoluble. This carboxypeptidase A, insoluble fractions. What about alpha tubulin? Well, that's not a secreted protein, and you know what? It doesn't matter. There's no insoluble. That means it's not an autophagy of the cytoplasm. It's an autophagy of the endoplasmic reticulum. That's where these proteins are made. Because remember, the secreted, and remember the exocrine function of the pancreas via the acinar cells, the ductal cells, is a secretion process. Just beautiful slides. This is just taking this data and making a histogram, right? And just showing you how much insoluble, lots of BIP insoluble, a lot of trypsinogen insoluble, a lot of amylase, a lot of the carboxypeptidase A. This means this stuff isn't getting out of the acinar cells. That means you got problems because of all this insoluble protein, right? If it's not soluble, it's not going to be secreted. So that's what this tells you. Perfect phenotype associated with trapping this gene in a mouse model. Very, very, very good research. Love it. All right. So let's do an analytical synopsis now of our paper. CCPG1 is a transmembrane endoplasmic reticulum, multifunctional, multi-domain polypeptide that's specifically transcriptionally hyperexpressed upon emblematic ER stress response. Sorry about that word as. I removed part of that sentence because it just repeated itself basically. Sorry. Now, the cytosolic N-terminal domain of CCPG1 interacts with those all-important linear peptides, LIR and FIR. Those are structural domains of the 2 key autophagy proteins, all the ATG8s and the FIP200, right? Very important. Remember those structures. Remember that huge bridge structure we talked about, bound to those phospholipids. The result of this protein-protein interaction, that CCPG1 with the ATGs and FIP, leads to autophagosome biogenesis. You know if you don't have it because you saw what happens with all those insoluble proteins. You just saw that data. So the pancreatic acinar cell, CCPG1 triggered endoplasmic reticulum phagy, maintains luminal rough ER proteostasis. So you want to have proteostasis, you want to have a constant amount of proteins moving through the system by putting the brakes on unbridled unfolded protein response signaling that could lead to tissue injury and death. So again, this whole CCPG1, it is the rheostat. It's controlling the amount of UPR you get so that you don't overextend the mark. Too much of that, you get too many insoluble proteins. Too many insoluble proteins, you can no longer carry out autophagy. If you can't carry out autophagy, the cell says, okay, I'm giving up. That's it, hands up, throw in the towel, right, in a fight. put up the white flag when you're at war. That means, okay, the cell's going to go boom, die, right? It's going to die. So you're likely going to lead to apoptosis. That could also cause an inflammatory response in the meantime. It could, because of necrosis perhaps happening instead of apoptosis. Remember those fine point signalings distal to the initial signaling? Or you can even get cell proliferation if you got the wrong kind of signaling, and that could lead to ductal carcinoma, another major fate here, if things are not controlled via this whole system. All right. So this leads to their, I think, I don't know, it's an okay slide. It's again from the paper. So you got all these insoluble proteins, ER stress, right? Yeah. So you get persistent stress, too much, death, kill it, right? Natural killer cells come along and kill these cells, or they just commit suicide. Now, if you are able to synthesize this discrete CCPG1 messenger RNA, you're now going to embed that protein into the phagosome complex, into the ER basically, right? And that's going to bind to the LIR and the FIRs. The LIR is part of the LC3 complex. The FIRs, the FIRs, are part of this whole FIP200 system, combined, because these are N-terminal proteins on this CCPG1, remember? You're going to then generate this autophagosome phagy. This whole complex starts to form, and you get the right thing happening. That whole system will help reduce ER stress because you're eliminating all of these proteins that are starting to go from soluble to insoluble because you're backing up this translocating loading process of proteins leaving the ER, leaving the cell. What cell? The acinar cell of the exocrine pancreas. Right? So it's a wonderful system to control it. And CCPG1, if it's not there, you don't get the complex, you don't make the autophagosome, and basically you go to death. Not good for the acinar cells. So there's a dialectic of all of this, right? Premises, or that is thesis, antithesis, synthesis, right? That kind of dialectic. Way far away from opinion, right? Pancreatic, so this is what we're doing now. We're basically verifying, VerEvMed, we're verifying the evidence here by carrying out a dialectic, which the paper provided for us to do. So dialectic says, from the research data from this paper, developmental cell paper, that pancreatic acinar cells become the initial target of pancreatitis-linked ductal carcinoma. We already know that, okay? That is a well-known thesis. Again, I had an entire, well over an hour lecture on that topic. So pancreatitis, which is nothing more than inflammation of the pancreas, can lead to just necrosis, which is certainly not good, but that comes from uncontrolled endoplasmic reticulum-associated degradation and UPR, uncontrolled. None of that's good, because let me tell you, the etiology of PDAC, which is pancreatic ductal adenocarcinoma, is first you have pancreatitis, then you have PDAC, then you're dead. Dead is the whole human, right? So all of the stuff you want to prevent. So having a functional autophagy system because of the CCPG1 complex and all those HG proteins and those really important phospholipids, right, which is another topic of a conversation, of course, because lipids are the most important thing here. But I let them play the back burner because I wanted to, let the proteins have their time because this paper was about a protein, CCPG1 and all that. All right. So excessive inflammation can lead to cell transformation, as we've talked about many times in these lectures. And finally, if that happens, pancreatic ductal adenocarcinoma, very deadly, 100% aggressive, ultimately lethal cancer in humans, right? Tombstone stuff for real. Very bad stuff. Very sad stuff for families. Therefore, CCPG1-linked autophagy, which maintains proteostasis. Proteostasis means keeps the protein levels constant of the acinar cells, which again are those exocrine cells for the duct, can block transformation to PDAC. That's a really important thing. That's why this paper made it up in Developmental Cell. It means that, yeah, sure, pharmaceutical industry is going to be really interested now in CCPG1. Right? It's going to make sure the CCPG1 is functional when it needs to be, so it maintains proteostasis. It doesn't allow ductal cells to go into a fate, a destiny that will lead to a fate that we don't want, such as necrosis, inflammation, like pancreatitis, and then ultimately maybe even adenocarcinoma. So thanks a lot for paying attention during this talk. I know it was more complicated than some I've given because we got into the molecularity of a very specific pathophysiology. We also were able, though, I think, go into what autophagy is. And particularly, we jumped right into ERphagy, which hopefully now you have a really good understanding of. And you know why it's so important. It's not something esoteric. As a biomedical person, as a health science, as a healthcare provider, as a healthcare scientist, as a biochemist, as a molecular biologist, as a veterinarian, as a pharmaceutical scientist, and even somebody who works with law associated with drugs that may impact autophagy in cells, those ones in cancer therapy, for example. This is good stuff to know. And I can give you a lot more information about it. All you need to do is contact me, and I'd be glad to take you on as a client. So there is my specific email address. You can go directly to that. I do respond to my email address within 24 hours. You can go to my Facebook. You can check out this talk, which will be posted there very soon today. Today's the 12th of February, by the way. That's Abraham Lincoln's birthday. We used to get this day off in Chicago because Abraham Lincoln was supposedly from Illinois. Really, he was from Kentucky, but we're not counting Kentucky. At any rate, because he was a senator of Illinois, I think. At any rate, we're at VerEvMed. Here's our email address for VerEvMed. Contact us through that, and you can become our client. Just go to the website and see all the great things we offer. Remember, VerEvMed is verifying published evidence in medical biosciences. So I want to thank you again for your attention, which I presume was there. And I want to say thanks for contributing to VerEvMed and to the success of VerEvMed and to overall program that we're trying to generate here. Again, I'm Dr. Dan Guerra signing off from VerEvMed Studios in the Pacific Northwest. Until next time, bye for now.